Well, welcome back. It's really my pleasure to have this chat with Dr. Amy Abernethy. I'm going to tell you a few things about her, but I'm not going to be my usual inappropriate self and instead I'm going to refer you to a great chat that she had in Tectonics with Lisa Suenen's podcast, where she really talks about how she got to where she is. And I thought I just learned a lot about how wonderful Amy is as a person and where she comes from. It's really it's heartening when you see where our leaders come from. So I strongly recommend that podcast. So Dr. Abernethy is our Principal Deputy Commissioner and Acting CIO at the U.S. Food and Drug Administration. She's trained as a hematologist and oncologist and palliative medicine physician. She's a clinical data expert and a clinical researcher. She really does understand clinical data, and she understands both sides of this road. She served as Chief Medical Officer, Chief Scientific Officer, and Senior Vice President for the very successful company Flatiron Health, which was recently acquired by the Roche Group, where she led research oncology. So thank you, Amy, for making a little bit of extra time. I promise to get you out on time. I'll interrupt us at around 12:30 to get some audience questions. But here's my first question for you. I think we began to already talk about frameworks for therapy, and I think you've already begun to answer the question of how do you scale up, which is reorganizing the enterprise and creating these frameworks and these checklists. Does that give you optimism that the FDA is going to be able to keep up with the oncoming onslaught?
So first of all, Zach, thank you for having me here with you today. And also, thank you for the moment to reflect in the previous 8 minutes and 46 seconds. It was really important. And your question was around this world of hyper-individualized therapies and do I think the FDA is going to be able to keep up? You know, first of all, as FDA, I think that we do already have the appropriate authorities in place to both support the innovation in the space of hyper-individualized therapies and regulate them. What I mean by that is I don't think we need to go and ask for new laws and a new rule set, but rather we can work within the laws that we already have. You already heard me speak about the concept of defining our regulatory thinking through frameworks, and I think that's going to be an important area that's going to continue to happen. We do that by also getting lots of stakeholder input. And in March, just before all the activity going on with the pandemic, we held a stakeholder meeting to get input to guide our thinking in this space as well. And many times our thinking is then codified in guidance documents, and I can't really state when such guidances will come out, but I do reflect on the fact that we have been putting out a lot of guidances as it relates to cell and gene therapies this year, and it had a package actually of some of those put out in January. So I think you'll continue to see development of our thinking in this space. But also, is it going to be possible to regulate therapies that may be appropriate for only one or several people, one therapy at a time? And I think that there are additional mechanisms or maneuvers that could potentially help. So for example, the development of platforms where specific components of the development of individualized therapies have already been fully approved, complete with CMC documentation and understanding of both the preclinical and maybe even some of the clinical evidence to go along with it, so that information for the first sort of platform-based, maybe that's a workhorse cell or sort of a general approach, can now be a part of the development of now a customized therapy that sits on top of that platform. And I anticipate one of the ways that this whole space is going to scale up is through the development of kind of these platform-based approaches, and that's also going to help us from the regulatory side because it'll certainly streamline that. I think another way that you can anticipate seeing streamlining in hyper-individualized therapies is for example, streamlining of submission documentation and sort of what we expect to see, which will go along with the regulatory framework. Practically speaking, we have a long way to go together as a community, and I think there's going to be a lot of work to be done. But you know, as there's practice, practice leads to standards. As there's standards, there's much more effective and efficient communication.
I think that's right. I think that the practice is what's going to get us there. So you really did innovate a lot about this whole gathering data to understand the effective therapies in your previous role at Flatiron. And we've all heard probably ad nauseum this the problem that clinical trials are expensive, they're long, and we'd love to be able to do much more of them for every single question, but they're also very expensive and they're slow. At the same time, we're quite aware that observational data sometimes is a poor substitute for clinical trials. And I wonder how that makes you think about, is there any innovation in that space that we can do? Anything that you can think of from your Flatiron days that might actually inform, maybe not today, but tomorrow, what the FDA does?
So in my past life, I went out on a limb and I highlighted that in the 2000s, we talked a lot about big data science leading to comparative effectiveness research. And we predominantly talked about it with respect to antihypertensives or lipid lowering agents. And in fact, when I said I went out on a limb, I actually highlighted that I thought that the real place we were going to see real world data and sort of big data make big movements was in personalized medicine. And some observations I had during that period of time. One is that in rare diseases, in the personalized medicine space, we're going to need to rely on natural history to tell us the benchmarks, so to speak. You heard in the prior session around the kind of idea of developing synthetic control arms. Either way, let's talk about high quality natural history data and the ability to now bring in data from multiple different data sources, link that together to tell complete longitudinal case histories. So look, the second thing I would say is it's not just that it is about the benchmarking, it's about being able to assure longitudinality despite crossing health systems, despite maybe coming into contact with the health system and maybe getting care at home, all of those different features that I think is going to be important as we think about the role of real-world data. The second thing that I think is really important in the precision medicine space and a key innovation is a paper that I wrote about 1.5 years ago that I called Small But Mighty. And that's that oftentimes the data sets for real-world data in support of precision medicine questions, the data sets themselves are quite small. It might be around a cohort of 30 or 60 patients with a disease. One of the reasons I called them small but mighty is even though the cohort may be small, critical was the need to get deep and longitudinal, as I just said, but also ideally using appropriate privacy controls, etc., when needed to be able to go back and read the case notes or the narrative to actually fill in an understanding of the story and almost a qualitative plus quantitative kind of way. And those small but mighty narratives actually help to understand really what does natural history look like as we think of it benchmarks. The third thing I would mention as it relates to some lessons learned around where real-world data can help us with respect to precision medicine and individualized therapies is as we think about needing to get to these really unique cohorts that help to define precision medicine, we can actually use artificial intelligence and different maneuvers to get to really well-refined cohorts. So essentially using new capabilities for case definitions that allow for very well curated cohorts where we know that the right people are included and people are not being systematically missed and being able to measure that. And then the last thing is real-world data to do all the different things that we need to do as it relates to informing clinical trial design, informing, for example, longitudinal follow-up even after the trial has ended, and certainly helping us understand in the long term with these therapies, how do we worry about issues such as long-term adverse effects, late effects, etc. So I think there's a lot of places where real world data does play a role and complements what we're going to learn in clinical trials. It's not an either or. These pieces need to come together.
Well, I think people should really be carefully listening to what you're saying, because what you're saying resonates exactly what you said before about Julia, Mila's mom. By taking a careful history of the seizures, in this N of 1, we could see what was going on. And so if you're in that situation, taking high quality longitudinal observations is key. I want to bring up another really good paper that you wrote, which is your paper about where you showed that there perhaps are fewer mutations that really inform us than there are sort of treatments out there. And you can answer that, you can reflect on that either way you want, but one way you could reflect on it is, when we do trials of cancer drugs, should we just be looking at a few mutations, some gene panels, an exome, or a whole genome?
So that was a paper that when we wrote it created quite a stir, as you may recall.
I loved it.
Oh, a little bit of mix up, right? But, you know, practically speaking, I think the question becomes, you know, essentially, how do we right size sequencing clinically now? How do we think about its role into the future? And, you know, we kind of just touched on value a few minutes ago. And how do we think about the decisions that we make about right sizing sequencing with respect to the value equation? Now, I'm going to punt a little bit and say this is how it should be, because these days I'm a regulator. But, you know, practically speaking, the way that I have thought about this is that there is the information that's clinically actionable now. There's the information that also sort of potentially points the way to the future. So, you know, prepares me and my patient for what may come down the pipeline, right? And then there's also the information that has service beyond this particular patient, but may actually have a role, for example, as deep biological information in real world data sets, because we have that information that can be part of those data sets. And each of those has a different value equation. Probably, you know, really broad sequencing right now for the patient sitting in front of me, I probably don't have enough therapies for, you know, all the potential mutations that are identified. So it's just not fully actionable. If the person may live long enough, it may make sense to have that additional detailed information now, because as new therapies come down the pipe, that may be appropriate, and the clinician and the patient together needs to make that decision. And I certainly see a societal value as it relates to the research opportunity. And there, again, there's a value equation that's got to do with who's paying for that downstream value.
Well, let me pivot a little bit to what I started in my introductory remarks to talk about, which was COVID-19. And all of a sudden, we have this huge threat that we want to respond to. Initially, had very little data to go by, not enough data sharing. And all of a sudden now we have, I can tell you that in the most prestigious journals, literally 100 to 200 submissions per day of manuscripts. And I believe there's over 2,000 trials underway in treating COVID. So question I have for you is, what's the FDA doing to try to bring some sense to this ferment?
So first of all, I think we should be energized by the fact that the scientific capabilities can spin up fast enough to start to move. I'm going to come back to a but in just a moment now. It does mean at FDA, we've had lots of incoming INDs for repurposing of drugs that are already marketed and new drugs in development that may be pulled into the COVID-19 story. And, you know, we have been working as quickly as possible to process those INDs and get those clinical trials going. I think it is important, though, that just like in any other part of science, we prioritize getting the right work done first, second, third, so we can kind of essentially stoke the development of a pipeline. But as FDA, we've made a commitment for all hands on deck. Let's get it done. And we're processing through the IND, in the pre-IND meetings and the INDs, et cetera. So I think that you'll see that continues to flow through. Where appropriate, we have also utilized other actions such as regulatory flexibility. So, for example, what you've seen on the diagnostic side as it relates to EUAs, we have also tried to leverage COVID-19 pandemic-specific guidances appropriate for this moment in time that does allow for flexibility. So we've done a lot of things to try and right-size the regulatory lift to focus on the things that are going to be most important for both treating COVID as well as keeping the population safe. And then we'll need to adjust that across time. Now, you brought up a really interesting point that I want to hit upon, which is all of the articles coming in to the journals, because practically speaking, those studies that are coming in that are well done clinical trials, randomized, hit their sample size, there are some, and they're actually starting to happen more and more. But the truth is that's not the majority of the information. And a lot of what we're seeing is the analysis of electronic health record data, or sort of small cohorts where people are trying to make assessments that have clinical impact, but maybe not based on credible evidence or sort of the best done methods. We'll just say it that way. And I think we can all just kind of understand what I've just said, and we'll leave it at that. And so one of the things we've really pushed on at FDA is something we call the Evidence Accelerator, which is a partnership with Reagan-Udall Foundation and Friends of Cancer Research, who are managing or running the Evidence Accelerator, and we, as FDA, provide technical assistance and scientific expertise. And the idea is to create a community of data holders and analysis teams working on COVID-19 with urgency, where we're developing the right methods together to analyze real-world data. We're creating communities and fora, like what we call lab meeting every week, to cross-check each other's work and provide real-time peer review, and where we're continuously analyzing data sets using common protocols and focused on common questions with common data elements in order to start to create some order around all of these kinds of findings within the real-world evidence space. And part of the goal here is to actually start to figure out how do we judiciously use real-world evidence and real-world data for COVID-19. How do we develop the right methods, and how do we prepare for the role of real-world data and real-world evidence in the future as the COVID-19 story continues to roll out?
So, Amy, that sounds really interesting. For enthusiastic, especially young people who want to get involved, is there a spot that they can look at the FDA website so they can see what's going on in that regard, or how to get involved?
So the Reagan-Udall Foundation is the foundation that's congressionally mandated and sits just next to the FDA. If you Google Reagan-Udall Foundation and COVID-19, this will come up. Otherwise, the website is www.f, as in friends, ocr.org/COVID-19.
Great, thank you. That's extremely helpful. Because we're running out of time, I'm not going to be able to ask you perhaps the most important question, which is how do you become a great Amy like you? So for young people who are in training, and they do ask me all the time about career advice. So I guess I'll have to be for another time, but it is important. But I do want to let you go at 12:45 as I promised, and so I want to go to the audience now. Now, as before. We're going to have Slido presented questions, and the eminent Professor Matthew Might is going to be reading out the questions. Thank you, Matthew. Matt, I don't know if you heard me at the beginning, Matt, where I gave your great quotes from your first keynote in our first session. But while we're doing it, let's exercise our Slido skills. Take it away, Rachel, please. Not bad, 100%. You guys don't know the trick questions when you see them. Now you're talking. All right, you're cued in. All right, excellent. So I hope your questions are appearing. Matt, take it away.
Okay. Will the embrace of hyper-individualized therapeutics at FDA shift the regulatory focus and burden from safety and efficacy to just safety? And if efficacy will remain a metric to reach patients, how will FDA evaluate efficacy before treating a patient with a custom therapy?
So first of all, I can't predict into the future and give us this is what it's going to look like in the future, but I can highlight a couple of things. As we talked about in the prior session, certainly evaluation of efficacy was one of the key elements of the framework described in the New England Journal editorial by Janet Woodcock and Peter Marks back in October. But as was just mentioned by Zak, that efficacy evaluation was really based on Julia's careful diaries and understanding the prior experience with seizures and then being able to describe the impact of the therapy based on individual experience. And so I think that story will be an important story that helps to understand what this may look like in the future. That being said, our public forums, stakeholder meetings, et cetera, will be an important place where this conversation will continue to play out as then it ultimately moves to how do we think about efficacy and the guidance as it gets developed around it.
Okay, and somewhat related to that, are there plans to hyper-individualize the regulatory approval for administration of hyper-individualized treatments? For example, would severity and illness of potential treatment toxicity serve as parameters to shift regulatory approval thresholds for individual treatment?
So I don't know where that goes, but that's a great suggestion to recommend. Whether I'm not sure if there's a public docket open right now, but there's always ways to submit your suggestions to the FDA through the docket process. So I recommend doing it.
By the way, I apologize all. I'm told by the word from above that we have to end at 12:40. So we have 7 minutes.
Okay. And does the FDA have a plan to approve every hyper-individualized treatment? How far does a physician's discretion go under practice of medicine when it comes to administration of a hyper-individualized treatment?
So with respect to the plan to approve every hyper-individualized therapy, I really sort of need to leave that to our biologics center, which is responsible for really thinking about how we are ultimately going to develop and apply the frameworks at scale. And I really do recommend kind of continuing to watch this space and also become part of the conversation. The other part of this was about physician discretion under practice of medicine. And I think that physician discretion under practice of medicine will continue to be managed and maintained similar to as we do with any therapies. And I don't expect that to majorly change specific just hyper-individualized therapies. We think at FDA about regulating medical products, not about regulating the actual practice of medicine. Those two things are as you know, quite distinct.
Okay, and then we have a COVID-related question. On approaching real-world evidence on COVID-19, you have 3 buckets of questions. I wonder what has been done for the first bucket, that is questions that can be answered within 2 weeks because the data are available. So do you happen to know what's been done with respect to COVID-19 on that bucket of question?
So great question. So what this is highlighting is that within our evidence accelerator, one way that we divided up the questions was those things that could be evaluated in, essentially 1 to 2 weeks. Those things could be evaluated in 3 to 12 weeks and 12 weeks plus. And that was for 2 reasons. One is the urgency of COVID-19. What can we do right now? And 2 is help to sort out what data sets are already curated and ready to go because we could start to understand who had data sets to answer what kind of questions in time periods, and then what was it going to take to curate and figure out methods for more complex questions. So this bucketing of questions into time scale, what served both purposes. In terms of what kind of questions that have been answered in this sort of first bucket, most of them are operational but critical. So for example, helping to understand what are the patterns of drugs for patients with COVID-19 in the ICU in terms of what they're receiving, for example, pressors, management of dialysis, etc. So what's the patterns of therapy? What are the drugs being received? And how does that help us understand, for example, surges in drug demand as more and more patients were in the ICU, better management of product supply chains, and planning for potential shortages. And so that's an example of the kind of like 3-week question that we've been dealing with. Another one has been around anticoagulants and understanding the use of heparin, low molecular weight heparin, and how that's changed over time. Another one has been around understanding, for example, what has been the impact of the COVID-19 scenario on cancer screening, including breast cancer screening, colorectal cancer screening, and what does that tell us about what potentially may be coming down the line, not for the patient with COVID-19, but for patients who have not had access to care. So we've shown a lot of questions that have fallen into this category that's helped us think about and plan as a community.
Another COVID question. So building on that, what have we learned from COVID-19, this COVID-19 experience that will survive COVID-19 and then apply to other disorders?
Oh, we're still learning, right? We within the context of the COVID-19 experience, I think we're learning in a number of different categories. So one is within the category of what I'm going to call regulatory flexibility, and what is our different elements of regulatory flexibility taught us about what regulations may look like in the future. And let me give you sort of 2 examples. One practical example is the use of telemedicine as remote visits within the context of clinical trials. That was already something being contemplated and described in our guidances, but not well used. And we're learning a lot about telemedicine, data quality within the context of remote visits, and that will help us long into the future. Another aspect of regulatory flexibility is what we learned in diagnostic tests. And as we had more regulatory flexibility, we started to see, for example, serologic tests not performing as well. We need to sort of back off with that regulatory flexibility and add in some requirements about submitting specific data around sensitivity and specificity of serologic tests. So we learned kind of what happens in the market and how to right size that. So that's one area of learning. Another area of learning is ultimately, how do we quickly marry what we know from the side of biology with what we're learning in both real world data and clinical trials to start to figure out how do we sort of prioritize and think about therapeutic development. And one practical example that's sort of off market, so it's easy to talk about, is the role of anticoagulants. And so, practically speaking, we're learning a lot on the biologic side around how COVID-19 may activate the coagulation cascade. We're learning a lot on the clinical side. That now teaches us both what clinicians are doing in the ICU, and we can learn from that, and then take that information and design clinical trials in a very thoughtful and more precise way. The last thing that I would say is we're learning a lot on the real world data side around what's the methods to do this quickly and at scale. How can we use new and novel data sources that we haven't previously really leveraged at scale? Everything from sort of hashing technologies and standardized analytic platforms to, tomorrow at lab meeting, we're going to be talking about synthetic data. I don't know where that goes from a regulatory perspective yet, but it certainly is pushing us to think about what all can we kind of really tackle on the real world data side that will help us with COVID-19 and into the future.
So thank you, Matt. Thank you, Amy. We're reaching our time. Amy, I want to tell you, in that last survey of all the lessons learned, you really showed what a synthetic leader you are. It's really, really impressive. I think we're very lucky to have you at the FDA. I think we're all grateful. I certainly am grateful. And I'm also very grateful that you spent some time with us today. So thank you very much.